Hello, and welcome back to Introduction to Genetics and Evolution. We talked in some of the previous videos about mutations and about some of the complications beyond standard simple single gene inheritance. What I'd like to do now is come back to mutations just briefly, talk about mutation rates, or essentially how often do mutations happen. Like I mentioned briefly previously that they happen a lot, but let's actually put some numbers on this. Well, again, let me reemphasize all genetic variations started as new mutations. Now, there are two questions people often want to know. There's actually a third one that we won't talk about, which is how often good mutations arise. But people often want to know how often do any sort of new mutations arise? How often is there any sort of alteration to the genetic code? Like, do you, for example, have any mutations relative to your parents? Will your kids have mutations relative to you? And then related to that, how often do bad new mutations arise? Do you have any bad mutations relative to your parents? Or will your kids have some bad mutations relative to you? Theoretically, we could do the same thing with good new mutations, but they're so infrequent and hard to measure that we probably can't do that very easily in this context. Now, how do you measure a mutation rate? So how do you measure a rate of new mutations? Well, mutation rate is typically measured as per generation and often per base. But again, measuring this, you have to define your parameters very clearly. You have to standardize by unit of time, which is typically the per generation. And you have to identify what kinds of mutations count. Like, are you identifying the mutation rate from one specific base to another specific base, like the mutation rate of a particular site from A to G in particular? Or do you mean just from that base to any different base, from A to anything but A? So this would include a mutation to G, a mutation to T, or a mutation to C. The other thing, too, we've often talked about this in the context of DNA sequence, but there are other kinds of genetic mutations, too. There are things like inversions. This is when you have a stretch on the chromosome flip, literally like just completely reverse its order. So all the same nucleotide sequence is there, but the order of it has changed. Or translocation, something where a piece goes from one chromosome to another. Or deletions, just a segment of DNA that just gets erased out and then the two ends get stuck together. Do you count those or do you not count those? And what people traditionally did, if you go further back in time, is people would measure mutation rates in the context of anything in a gene that yields a phenotype. Now, this, of course, ignores a lot of neutral changes and things like that. So you have to define what you're talking about. Typically speaking, people tend to focus on this when they're looking at a mutation rate from a specific base to any different base. So let's say, for example, that you know it at one particular site you started with an A, that when it changes to something but that. Okay? So we'll use that for the rest of this lecture. So how do you get this mutation rate per generation? Well, again, as I said, we'll use this base to any other base definition. And what people often do is they'll get a sequence of either a whole genome after a known number of generations or a big stretch of DNA. So let's imagine this is a 10-base genome. This is obviously ridiculously small. ACA, GG, CATTT. Okay, and after one generation, you look at the offspring and it's ACA, GG, CAT, CT. So what do we have here? We have one mutation, right? How many bases do we look at? Well, we looked at 10 bases. And how many generations? We looked at one generation. So in this case, the mutation rate would be 0.1 mutations per base pair per generation. So that's the kind of thing you can do. This is something that's commonly done in model organisms, such as, for example, C. elegans. This is a classic worm example. Now, another thing people will do is to compare sequences from existing species and estimate the number of generations. So, for example, if you could get a guesstimate of how many generations there are that distinguish humans from chimps, and you counted all the base differences between humans and chimps, you could get a mutation rate from that as well. So let me give you a couple of real numbers. This is an estimate of a mutation rate from worms for these C. elegans model systems. So it's 2.1 times 10 to the minus 8. This sounds very small, but trust me, it's bigger than it seems. 10 to the minus 8 mutations per base per generation. Now, what would this actually mean if you apply this mutation rate to humans? Well, the human genome size is 3.1 times 10 to the 9 bases. So how many mutations, if you just extrapolate directly from this mutation rate to this genome size, how many mutations on average would you expect individuals to get in one generation relative to their parents? The answer from this is 65. So that is not a small number of new mutations, right? And as I said, mutations are happening all the time. And amazingly, this finding is absolutely confirmed. That you see a 2012 study from various Icelandic families found on average 63 new mutations. This shows the value of extrapolating from model system studies. That estimate of 65 came ultimately from the studies of C. elegans worms. And from that, I just did some math to calculate what we would expect to see in humans. And in fact, we see a very, very, very close number to what I predicted from the C. elegans studies in humans. Now, interestingly, I didn't mention this before, there's a very strong effect of father's age. Now, most new mutations that humans get tend to be from their fathers rather than from their mothers. The reason for that is because in the production of sperm, you have many more cell divisions than you do in the production of eggs. So there's many more opportunities for an error to come in or for a mutation to come in. And in fact, what you see looking at this, there's a very strong age effect of the father. So here on the x-axis is the age of the father at the time of conceiving the child. On the y-axis is the number of new mutations found in that child. And what you see is there's a very dramatic increase as the father goes from 20, 25, 30, 35, 40, you see way, way more new mutations in the kids. In fact, for every year of the father's age, you see, on average, about 2 more new mutations. And this holds up with disease studies, that people see higher incidence of diseases like autism and other things like that in kids from older fathers. So this is something to consider, and it has definitely societal implications as well. So, coming back to what I said before, your brother is a mutant, but sadly, so are you. So you can call him a mutant, but he can call you one as well. But very importantly, these new mutations arise and they introduce a lot of variation into natural populations. And this ties back in with the previous video I just emphasized to you. Now again, what happens when these new mutations come up? If it's a new mutation, it's not been found before, then you have the birth of a SNP. You have this new sort of variation out there. Now these new mutations may spread, they may be found in other individuals, they may be lost by natural selection because it's bad. If it causes instant death, obviously it'll be lost very quickly. It may be lost from other forces, it may be lost just from a random event. That again, the individual with a new mutation gets hit by a truck when they're still young. Now while they're present, these new mutations, these new variations will have a frequency in the population. And this is something that will be very important when we get into population genetics, that alleles are present at a frequency within populations. Now you can see this as an example. Here are allele frequencies of blood types in the United States, just as an illustration. About 24% of the alleles out there are A, 10% are B, 66% are O. Okay, so if you took just what's going on, it took just everybody's chromosome and just lined them up, you would count on average these numbers in the United States. Now these are the allele frequencies, but there's also an underlying genotype frequency, right? That a subset of these individuals have to be big A versus, well, it's obviously big A. AA versus AO, a subset are AB, a subset are OO, etc. So you can put all these things together, and these genotypes, the fraction of individuals that have a particular genotype, have some sort of frequency in the population, and that is what is studied in the field of population. We'll come back to that far more extensively in later videos. But again, all genetic variations started as these new mutations. Now, the tougher question is, how often do bad new mutations arise? Well, there have been estimates of this as well. The deleterious mutation rate is, again, fairly high. And if you look at chromosomal anomalies, things like, just severe chromosomal disruptions, these happen quite a bit, and they probably have a very immediate effect on offspring and result in miscarriage. So as many of you know, on the order of about half of pregnancies are spontaneously aborted. It's estimated that maybe around half of those ones that are spontaneously aborted had some sort of genetic reason for it. They had some sort of chromosomal anomaly. They got too many copies of one, or things stuck together that weren't supposed to stick together, things like that. These are ones that you can't actually measure because it happens so frequently, there's no way to tell. But a lot of other bad mutations are far more subtle, right? They don't have such severe effects that they just make an embryo die, or they're masked because they're recessive. Well, current estimates suggest that on average, and this is true for humans as well, there are 1 to 2 bad new mutations per generation, per individual, right? So I'm sorry, but you're probably a bad mutant too. But I hope you'll come back nonetheless and watch the next video. Thank you very much.